In our DNA, we have thousands of genes which encode various traits from simple traits such as eye color to more complex systems such as vision. To create this variety, there are multiple versions of each gene, and we call these alleles. These versions encode different physical characteristics or phenotypes. While multiple alleles can exist for a particular gene, for example, a brown, black, or red allele for eye color, a diploid individual will only have two alleles. These alleles may be the same, meaning the individual is homozygous, or different, meaning the individual is heterozygous. Not only can mutations in alleles affect phenotype, but mutations in distinct genes can also result in the same mutant phenotype. Therefore, how do we determine if certain alleles encode the same gene or different genes? In this video, we want to briefly go over a common technique used in genetics labs to answer this question, the complementation test.
More specifically, the complementation test allows us to determine if two individuals with the same phenotype are genetically identical, meaning they have the same allele mutation at one particular gene, or are genetically different, meaning they have mutations at different genes. In a complementation test, we can cross two individuals with the same mutant phenotype and observe the phenotype of the progeny. Let's take a closer look at the genetic basis of this test to better understand what the progeny's phenotype can tell us. If we cross two individuals who are homozygous recessive for their mutation, then they will each donate one recessive allele to the F1 progeny. If these two recessive alleles are of the same gene, then the progeny will have the same mutant phenotype, and we refer to these genes as non-complementary, or genes which fail to complement. However, if the two recessive alleles are of different genes, then each parent will donate one wild-type allele for the other parent's recessive allele, and the progeny will have a wild-type phenotype. These genes are thus said to be complementary. To better visualize this, let's use the example of two mutant flowers. In our example, wild-type flowers are purple, but the two mutant flower strains are both homozygous recessive and white. We want to know if these two flowers have mutations in the same gene. Let's call this gene A with possible alleles big W and little w, or in two different genes. Let's say gene A with allele variants big W and little w, and gene B with allele variants big X and little x. If the two flowers both have mutations in gene A, then they will each donate a recessive allele little w in the cross, resulting in a progeny with little w genotype and a white mutant phenotype. However, if flower 1 has a mutation in gene A and flower 2 has a mutation in gene B, then flower 1 will donate little w and big X, and flower 2 will donate big W and little x. As a result, the progeny will be big W little w and big X little x for the two respective genes, and this heterozygous genotype will result in a purple wild-type flower. In the first example, we would say that the two mutations are non-complementary. In the second example, we would say that the two mutations are complementary.
However, complementation tests can be used for much more than flower color and have important clinical relevance. For example, if we know a particular mutation results in a certain disease in a mouse, and we observe the same disease in another mouse, we could cross the two mice and see if the progeny is wild type or also has the disease phenotypes. Using the complementation test can thus provide us with more information about the genetic basis of different diseases.